My name is Clara and I'm going to be presenting our work from the Salk Institute on modeling the prefrontal cortex as a lifelong learning system. Just as an introduction, I am an undergraduate neuroscientist working under PhD student Kanha Batra at the Thai lab. Before diving into our proposed architecture, I'd first like to go over the definition of lifelong learning. It's defined by three primary postulates, including learning and retaining knowledge of multiple tasks, using or adapting past knowledge to facilitate faster or more efficient learning of a new task, and being able to seamlessly switch between multiple tasks. Imagine a world in which we can pool our resources together to build models that could generalize diagnostics and identify biomarkers across diverse populations without the need for huge resource-intensive data sets. We'd be able to improve communities without highly developed healthcare infrastructures and make healthcare AI more equitable. There have been many attempts at emulating lifelong learning in computational models, each having various degrees of success and precision. Today, I'd like to present a novel architecture inspired by the prefrontal cortex and existing computational methods. First, let's take a closer look at how the brain directly addresses lifelong learning in the PFC, which is a brain region implicated in higher order cognitive functions such as social behavior and decision-making. Miller's classic example of context-dependent decision-making is how one might respond to a phone ringing in different environments. When you're in your own office, you instinctively lean over to pick it up, whereas if you're in somebody else's office or home, you'll wait for them to pick it up. According to Miller and Cohen's depiction of the prefrontal cortex, the PFC is like a switch operator in a system of railroad tracks, directing incoming sensory information, activating different pathways, and generating different responses. The PFC is capable of modeling context-dependent decision-making. As information enters the same sensory network, as sensory information enters the same network, but produces different responses depending on the track selected. Let's turn our attention to the center figure, which was created and published by a prior grad student in our lab, Caitlin Vanderweel. According to her results, one way the PFC might accomplish context mediation is through dopamine, which modulates the signal-to-noise ratio in subpopulations of MPFC neurons. Here, dopamine modulates the incoming signal in a context-dependent manner, amplifying the sensory response in response to an aversive stimulus, which is the air puff. Taking inspiration from our prior findings with dopamine, in a collaborative project led by Ben Suda from Terry Sanowski's lab at the Salk Institute, the classical mixture of experts framework was adapted to model the PFC's ability to encode multiple schemas. However, this solution has certain limitations. First, the task used to optimize this network was not rooted in the behavior of a real organism. And second, in adulthood, brains do not undergo the type of neurogenesis that can generate a new, distinct ensemble of neurons for every novel task. So, inspired by both the strengths and weaknesses of this model, we created a novel architecture. Our proposed architecture is a modular deep learning network called Neuromodulated Mixture of Experts, or NEMO. Now I'm going to step through a side-by-side analogy that'll show the biological inspiration for our architecture. So on a computational level, the supervisor network models the neuromodulatory inputs coming in from different parts of the brain to the prefrontal cortex. On the bottom is an example of a snippet of neuromodulatory signal from the prefrontal cortex. The expert networks shown in blue are recurrent neural networks modeling ensembles that are homogeneous in their neuromodulatory receptor distribution, and these ensembles form microcircuits in the PFC. These are inspired by cortical columns found in the brain, as they are considered functionally distinct units. The receptor network depicted in purple is a feed-forward neural network that models the sensitivity and specificity of neuromodulatory receptors. On a computational level, these can be interpreted as a sequence of gain modulators that are applied in a sequential order and dynamically to ensembles of neurons in order to optimize performance across different tasks. This is inspired by the concept of variable GPCR distributions in the cortex. Lastly, the inhibitory action selection component can be simplified to a version of the striatum. Modeled as a softmax function, it takes in the decisions from individual experts weighed by the neuromodulatory filters and combines them to make a final policy for the agent. It is inspired by the principle of mutual inhibition, which is found in the striatum. One of the most exciting features of NEMO is that we can model multiple timescales of information processing. First, on the top, we have rapid dynamics, modeled after neurotransmission that occurs on the order of milliseconds via ion channels. Second, we have a slower timescale, modeled after neuromodulation occurring on the order of seconds to minutes due to secondary messenger cascades. To test this model, we developed a novel multi-context foraging task where mice are presented with different environmental contexts indicated by different ambient lighting conditions. Each of these contexts, which we call seasons, correspond to different reward and cost contingencies. The mouse is placed in a chamber where there is a home area free of shocks and a shock where they must cross in order to receive a reward during a 20-second period indicated by a tone. To better understand how our paradigm works, let's take an example like summer. Summer will have a higher quantity of chocolate milk, which is the reward dispersed during the trial with less frequent shocks. And it might be indicated by a purple ambient lighting, but of course it's counterbalanced. We can look at the probability of a port entry across the trial. The x-axis in each of these plots begins 20 seconds before the reward tone plays, and those vertical lines indicate when the reward is available in the port. We collected data across 6 cohorts of mice, and an important takeaway from the port entry data is that there are reliably more port entries in summer than in winter, and our fall condition, which is a combination of the winter and summer conditions, contains more port entries than winter and less than summer, which is exactly what we would expect. These reliable and reproduced trends tell us that the mice have adapted their behavior to the specific contextual cues. Importantly, these probabilities were not independent of the order of context presented to the animals, showing that the learning occurring in the first trial might have an impact on later seasons. Given the context-dependent port entry data, we wanted to see if we could actually decode the context from these behavioral features. So we used SLEEP, which is developed by our collaborator, Telmo Pereira and Terry Sanowski's lab, to estimate mouse poses across videos and extract relevant behavioral features. After training a GLM on features extracted from the SLEEP output, we found that we were able to decode context from the features listed in this block, demonstrating the validity of the task as mice produce specific behavioral strategies for each context. Inspired by our lab's previous findings that dopamine contextually mediates the signal-to-noise ratio in MPFC subpopulations, we performed fiber photometry to measure the dopamine in mice during our task. Here we're looking at the fluorescence Z-score to 5 seconds before the tone. We see some preliminary results that the dopamine response in this region does have context specificity. And this figure shows the average pooled fluorescence per context. There seems to be a correlation in terms of the Z-score and the amount of reward present in a particular season. Next, we recorded neural activity in the MPFC using 1-photon calcium imaging while the mice were performing our task again. All of the rows in this plot correspond to individual neurons aggregated across multiple mice, so it's concatenated together. And using hierarchical clustering of the pooled neurons, we were able to identify ensembles of neurons that appear to have context-specific signal profiles. So as you can see, ensemble C on the right side seems to have a particular response to reward, whereas ensemble D seems to have a particular response to summer. Next, using a GLM trained on the dopamine sensor data, the model was able to successfully predict the context based on the signal for reward, summer, and winter, but the signal was not significantly decodable for fall due to possibly a lack of trials. Although this data is quite preliminary, we already see evidence of dopamine carrying information about the contexts. Unlike calcium, which has a finer granularity, dopamine is a single value corresponding to local activity. Next, let's shift our attention to the figure on the right. We trained a GLM to predict context from neural activity, and we found that the performance was significantly higher than a random controlled shuffle for each context. Together, these behavioral and neural findings suggest the presence of expert-like ensembles found in the MPFC that are modulated by neuromodulation and carry context-specific information. So the purpose of the preceding experiments is to understand the behavioral and neural implementations of lifelong learning. Importantly, they provide information for us to directly compare our NEMO architecture to real data. On the computational side, we designed a deep reinforcement learning simulation using the NEMO architecture. Essentially, a simulated mouse receives a simplified approximation of what mice in the real behavioral task would receive, such as tone, context cues, reward value, and shock value. The environmental inputs are pushed into the network, and the network uses several layers of processing and decision-making to generate a go or no-go behavior, modeling the port entries of the mice. So the simulations I'll be showing you right now have 2 neuromodulators and three experts, where each simulation can be thought of as individual mice. First, this is the example simulation. As the agent learns, the neuromodulator weights change across the trials. Eventually, the strategy stabilizes at the 30,000th point for both neuromodulators. And at this stability point, the experts carry different weights for each context. And you can see that in the pie charts. The neuromodulatory signals carry information about the context as well as the weights of the experts, which is exactly what we would expect based on our model. So if we take these results and look at the actual average go, no-go of the model, as the strategy stabilized over the course of the learning process, we see that the model converges to a similar policy as realized for each context, which is great. So to conclude, we are extremely excited by these preliminary results based on a binary simulation. However, we are also excited to expand our simulation to a 1D space and eventually create a simulation where we can collect rich information about neural data as well. Okay, so before I take any questions, I'd like to acknowledge every person from the Thai lab who's made this research possible, including Kay Tai, Romy Wickman, our lab manager, and Kanha Batra, our lead graduate student on this project. I'd also like to thank every member of the MetaMind team for making this possible. Okay.